We start with the chapter 4 on photosynthesis, and this is of the syllabus 5090 biology O levels. And this is, of course, it's a long chapter, so it probably go on to one or two videos. So this is the first video on it, and we are talking about this in January 2021, and this is for the 2021 syllabus. Let's look at the syllabus. The chapter 4 is plant nutrition. Plant nutrition is all about photosynthesis. So we're going to be studying photosynthesis in a lot of detail. As per O-level syllabus, you have to know the synthesis, the leaf structure, and the mineral nutrition. Then we will be tackling the first few points, which is the photosynthesis is the fundamental process by which plants manufacture carbohydrates. And then there are three practicals here, and then the equation, and then another three practicals here. So let's start with A, B, C, and D. And please, we will go back to this as soon as we have finished this, and we will refer back to it and talk about all the different syllabus points which we need to cover in this chapter. Now, the first point that we need to study is the equation for photosynthesis. Now, the equation for photosynthesis is CO2. Now, where does the CO2 come from? So let's look at a leaf. The CO2 is entering from the air through the stomata. So CO2 is coming from the air. Why is it diffusing in? Because it's in a higher concentration outside. And on a bright sunny day, it would be less CO2 inside the leaf. Why less CO2? Because it is being used for photosynthesis. Now the CO2 combines with water. Where does the water come from? Water comes from the soil, up the xylem, into the leaf. And then we say the carbon dioxide and the water combine in the presence of light and chlorophyll. And what does it form? It forms glucose, C6H12O6 plus oxygen. Now the oxygen is going to diffuse out. Basically why? Because oxygen is in excess, and I said it was a bright sunny day. So the oxygen is in excess, so that is going to diffuse out because some of the oxygen is going to be used for respiration. But because it's a bright sunny day, so the rate of photosynthesis is more than the rate of respiration. Please remember, respiration occurs during the day and the night. Respiration is something which keeps a plant alive. Just like we are respiring at all times, whether we are sleeping or whether we are up and about. So the carbon dioxide from the air combines with the water from the soil in the presence of light and chlorophyll. And what we don't mention here is the most important thing, the chapter which we just studied is enzymes. So all reactions take place in the presence of enzymes. Without enzymes, no biological reaction can take place. So what is going to be formed is glucose, and the oxygen is going to be the excess, so that is going to diffuse out of the leaf. Now a quick overview of the equation. This would be the chemical equation. Of course, enzymes is not written on it, but we have to understand is that enzymes are required for every reaction. And this is the word equation. You have to know both of them. And this is, of course, the symbol equation, and you have to balance it out if you're going to give me the symbol equation. So the formula for glucose is C6H12O6. It has carbon, hydrogen, and oxygen. So glucose is made up of carbon, hydrogen, and oxygen. And then, of course, this oxygen is a waste gas, so this is going to diffuse out of the leaf. Carbon dioxide is going to diffuse into the leaf, because if the rate of photosynthesis is higher than the rate of respiration. Then we look at the parts of a leaf. Petiole is the stem which attaches it to the main stem, and then this is the base, and these are the veins in it. This one here is the midrib, which is the main central vessel, and that's called the midrib. And these are the margin, and this is the apex, the tip, and this whole structure is called the lamina, or the blade. And this is a very large, a broad lamina. Why? Because more surface area, more surface area, more light absorbed. So we have to understand is how the leaf structure is adapted for photosynthesis, and we'll have to take a section of it from here, and then we're going to show you more of the details of that structure. This would be the structure of the leaf. The outer, this is the non-cellular layer. It's just like you laminate your copies or your books, cuticle. And then we have the upper epidermis. And then we have the palisade mesophyll. Now these palisade mesophyll cells are rectangular, and these have lots and lots of chloroplasts in them. And then we have the xylem and the phloem, which of course constitutes the whole vascular bundles. And then we have the spongy mesophyll, cells. And then the guard cells, and the stoma, which is the opening. So cuticle, upper epidermis, lower epidermis, palisade mesophyll, and spongy mesophyll. Now palisade mesophyll also has chloroplasts, spongy mesophyll also has chloroplasts, and the guard cells also have chloroplasts. So please, once again, we just do a quick recap. Palisade mesophyll has chloroplasts, more chloroplasts here. Spongy mesophyll has less of them, and the guard cells also have chloroplasts. So three structures have chloroplasts: palisade mesophyll, spongy mesophyll, and the guard cells. 1, 2, and 3. These have, so upper epidermis does not have any guard cell. No guard cells in the upper epidermis, no guard cells in the lower epidermis. No guard cells in the, no chloroplast in the upper epidermis, no chloroplast in the lower epidermis, and no chloroplasts in the, so this is what we have to understand. Upper epidermis, no chloroplast; lower epidermis, no chloroplast; cuticle, no chloroplast. So we have to understand the structure of the leaf and how this leaf is adapted for photosynthesis. Now, as we study photosynthesis, we come to a very general conclusion. What do you mean by photosynthesis? Photosynthesis means in the presence of light, what is happening? Carbon dioxide from the air and water are combining and is forming a glucose molecule, C6H12O6. Now basically what is happening is light energy is being converted to chemical energy. So this is actually transduction. So chemical energy is what? Chemical energy is this glucose molecule. And in the presence of light, the light energy actually converts a whole lot of reactions take place, the light dependent and the light independent reaction, which you will study in your A levels. And it helps in the combination of carbon dioxide and water and forms a glucose molecule. And we say light energy. Please remember, energy cannot be created or destroyed, and the law of thermodynamics say that. So is light energy being converted into chemical energy. So carbon dioxide from the air enters the leaf. The water molecule in the soil enters the root and comes here and comes up here and here. So the water from the soil reaches here, the carbon dioxide from the air reaches here, and this is combined in the presence of light to form a glucose molecule. But what we need to understand is that this glucose is the beginning of every molecule. From this glucose, we are going to make cellulose. From this glucose, we are going to make starch and store it in the cells. From this glucose, we are going to make fat. But the problem arises from this glucose, how are we going to make a protein or an amino acid? What we need is, we need nitrates from the soil. Now these nitrates will enter through the roots and come here, plus glucose plus the nitrates is going to form an amino acid. So amino acids can only be formed if there are nitrates present in the soil. And then these amino acids are going to be converted into proteins. So what we have to understand is that the glucose molecule is the basic unit which is needed, without which nothing can happen, without which this plant cannot grow. Cellulose, new cell walls have to be added. Just like if you need to add another room to a building, well, you need bricks to make the wall of the room. So cellulose, starch, store, because at night there's no photosynthesis and that starch is going to be mobilized. Then the fat, we eat corn oil, olive oil. That's when you crush the seeds and you get those fats. And then of course the nitrates combining to form amino acids. So the nitrates plus the glucose is going to result in the formation of amino acids, and these amino acids are going to be put together to make a protein molecule. And we need proteins, we need enzymes. Plants need lots of enzymes. Now, the practicals that we have in this chapter are the most complex and the most difficult ones. It says investigate the necessity for. Necessity means that it's absolutely essential that we need number one, chlorophyll, number two, light, and number three, carbon dioxide for the process of photosynthesis. Now, if I say necessity, it means that it is absolutely essential. Without any of these three, photosynthesis cannot take place. Now, for this, we need to do three experiments. Three experiments, one for each of these points, one for chlorophyll, one for light, and one for carbon dioxide. But before we start these three experiments, I would like to discuss two very important practical processes which we need to know before we start these experiments. The first one is destarch, destarching a potted plant. First of all, you must know why do we need to destarch a potted plant. So we have this plant and it is in soil, right? It's got roots and it's in this pot. Now, why do we need to starch a potted plant? We want zero starch because we're going to set up the experiment, and then we're going to say, okay, now this happens, so it is photosynthesizing, so it is producing glucose, and the glucose is being converted to starch. But before we start the experiment, we need to ensure that there was no starch present initially. So we keep it in a box, or you keep it in a dark place where there's no light, and you keep it in that for 24 to 48 hours. Now, during this 24 to 48 hours, what is going to happen? If there was any starch stores in it, the starch stores are going to be converted to glucose. The glucose is going to be respired and energy released, because this plant has to survive this time for 48 hours that you've kept it in a dark place. So de-starch a potted plant. That is what we write for each experiment, because we start the experiment when we set up the apparatus. We want to assume that there was no starch present in this plant initially when we started the experiment. So this is what we're going to talk about in every experiment. Then we're going to set up the experiment, expose it to light, and then we're going to detach a leaf. We're going to remove the leaf and we're going to test it for starch. Test this leaf for starch. Now, how are we going to do this experiment? How are we going to test a leaf for starch? Now, please understand this is different than if I had asked you test a piece of apple for starch. Now, we would do this very differently. What would you do is you would crush the apple. Why would you crush the apple? Why? Because we want to release the starch. We want to release the cell contents. So we'd crush the apple and we'd add iodine and we would see what is the color change. If the color remained brown, then no starch. If the color was blue-black, then starch present. But this is not the way. But when we have to test a leaf for starch, we cannot do it this way. There is a special way that we need to do this experiment, and you must know this, how to do the 4 steps in this test. So if I say test a leaf for starch, you're not going to give me the story about this piece of apple that crushed the apple, add iodine and brown and all this, and there's no heating in this. You're going to give me some other story, and that story is number 1, dip the leaf in boiling water. Dip the leaf in boiling water. Now, of course, you must know the reason why we are doing this. The reason why we are doing this is we want to stop all the reactions, so we say enzymes will be denatured. Enzymes denatured. Now, you usually say, some of you say, enzymes killed. I'm sorry, enzyme is not a piece of chicken or is not an animal which you're going to throttle and kill it. So, enzymes will be denatured. So, do not say enzymes killed. Enzyme is a molecule. So enzymes denatured because you've used boiling water and this temperature is very high, 100 degrees. So it's going to denature all the enzymes. So all the reactions are going to stop. Number 2, now you boil the leaf in ethanol. Boil the leaf in ethanol. Now why are we doing that is, please remember, ethanol is an organic solvent. So if you boil the leaf in ethanol, you are just decolorizing the leaf. Decolorize the leaf. Why we want to decolorize the leaf? Because when we add iodine, we cannot see the color change on a green leaf. So we need to remove the chlorophyll. And the chlorophyll dissolves in the ethanol. Chlorophyll dissolves in the ethanol. Like we say, sugar dissolves in water. But please remember, sand doesn't dissolve in water. So what you're doing is you're boiling the leaf in ethanol. And this, of course, decolorizes the leaf, and the leaf becomes absolutely white and it becomes very brittle. Brittle means it becomes very easily to break. If you just crush it in your hands, it'll just become in small pieces. Just like if you have these Lay's chips and you just crush them, it just breaks up into tiny little pieces. So it's very brittle. The word is brittle that we use. Brittle. If you don't know a word, please look it up in the dictionary. So we first dipped the leaf in boiling water. We stopped all the reactions, enzymes denatured. Then we boiled the leaf in ethanol. This will, of course, remove the chlorophyll and the leaf becomes very brittle. The third step that we do is you dip the leaf in water. Now this water is neither hot or cold, it's just ordinary room temperature. And this actually we do is to soften the leaf. Because you see, the why it becomes brittle is because all the water is removed, it gets dehydrated. So softens the leaf. And number four, then you place on a white tile, and you add iodine, and you add the iodine solution to it. Now when you place this on a white tile, the leaf is all just white in color, and if it turns blue black, that means starch is present. But if this leaf which you placed on a white tile turns brown, iodine is brown in color, and if it turns brown, that means there was no starch present in that leaf. So starch present, no starch present. So this is what we need to understand. This is starch present because it's turned blue black. So these are the steps in how we tested a leaf for starch. Dip the leaf in boiling water, enzymes denatured. Boil the leaf in ethanol, decolorizes the leaf, chlorophyll dissolves in the ethanol. And then we had dip the leaf in water, softens the leaf, and then you place it on the white tile and you add iodine solution, and you will find out if photosynthesis has taken place. Then glucose has been made, and the glucose has been converted to starch. So this is what we are trying to look at it. If we say starch is present, it means photosynthesis did take place, and we were correct in assuming whatever variable that we were testing. Now, the first necessity that we are going to study is we're going to study chlorophyll. So you take a plant with variegated leaves. Now, what are variegated leaves? Now, these are variegated leaves. Variegated leaves are which are naturally genetically in they show a pattern. They show some white areas and some green areas. Here also you can see some white areas. Now what you need to do is you need to make a tracing of this before you start anything on it. When we've talked about the practical, you realize why I want you to do a tracing of it. So you can see all these leaves have different areas which are white and different areas which are green. Now these are called variegated leaves. And you've seen a lot of these. We see a lot of these in Pakistan. And there are lots of plants with these sort of pattern. It's not that these are diseased or something, but they just have a certain pattern. Of course, some plants have totally green leaves, but these are plants with a special type of leaves which are called plants with variegated leaves. Now, the procedure that we're going to follow is first you take a plant with variegated leaves, then you destarch the plant, that is, you place it in the dark for 24 to 48 hours. Then you take it out and you expose it to bright light for 8 to 10 hours. Then you detach a leaf and you trace the leaf. Because what is going to happen is when you're going to test the leaf for starch, well, you're going to boil it in alcohol or ethanol, and all the green color is going to be lost. So you to just get a white leaf. Now you won't remember which part was green and which part was white. So in order to then see which part was green and which was light and which part has starch in it or not. So like for instance, this was the leaf and this was the part which was white. and this was the part which was green. Now you don't remember which one was green, so if you traced it out, then you'll know which part was green and which part was white. Because then when you'd add the iodine to it, then the green part will remain black because that's where starch will be present. And this part which will not which was white, this part will remain brown because the iodine doesn't have any starch. And this part will turn black, which had the pigment in it, the chlorophyll in it, which absorbs light and causes photosynthesis to take place. So this is why we said, and then you test the leaf for starch, and you find out the areas which were white actually had no chlorophyll. So no chlorophyll, no photosynthesis, no starch. This proves your point that chlorophyll is necessary for photosynthesis. Now, the next variable that we need to study is the presence of light. We say light is necessary for photosynthesis. So we do the same test, de-starch a potted plant, and you expose to bright light for 8 to 10 hours. But before you do that, you cover one of the leaf with black paper. So this one you cover with black paper, the others you don't. Now we cover the black paper on both sides. So this is the leaf that you've covered, this is the leaf that you have not covered. And you're exposing it for 8 to 10 hours to light. And then you detach this leaf, leaf A, and leaf B. And you test these both for starch. And you do the 4 steps, which is, of course, as I said before, dip in boiling water, then boil in ethanol, and then again dip in water. And then you add the iodine to it. Now, this leaf A, which was covered with black paper on both sides, received no light. Now, when it received no light, we find out that when you add iodine to it, its color remains brown. So that means no starch is present. So this again proves the fact that light is necessary for photosynthesis. Now, the last variable that we talk about is carbon dioxide. So we take 2 potted plants, same size, same species. We cover it with a clear plastic bag and we place a beaker in which the volume is going to be the same. In B we have distilled water, in A we have a chemical that absorbs CO2. So all the CO2 present here will be absorbed into this chemical, will absorb it. So this plant has no CO2, this plant CO2 has present, and we expose to bright light. So we expose it to bright light for again 8 to 10 hours, and then we take one leaf from here and we take one leaf from here and we test it for starch. So now as you know, this one did not have any carbon dioxide, so no carbon dioxide, so no starch. And this one of course had carbon dioxide because we placed distilled water here, so in this one starch will be present. And we will do the same 4 steps to test this leaf for starch, which we have talked about in an earlier experiment. So these are the 3 conditions which we say are absolutely necessary for photosynthesis, and that is number 1, light, number 2, carbon dioxide, and number 3, chlorophyll. So all these 3 factors, we've done 3 experiments. So you have to know one experiment, this, the second experiment, and the third experiment. So 3 experiments which prove that all these 3 are necessary for photosynthesis. And now this will continue on to the second video, which will complete this chapter. Thank you.